All right, hello everyone. I'm here to tell you about our shared resource, which is called PHISM, which stands for Preclinical Human Immune System Mice. And so it's easier, you just think of us as the humanized mouse core. You know, I want to start by introducing you to our team. My name is Julie Lang, and I manage the core and co-direct it along with Roberta Polenda, who I've been working with since 2008 on the humanized mouse model. And like all of you know, the most important part of our core is our technical team and the people, the expertise they provide. And we have a great team that I work with, including Matthew Lewis, who is a flow cytometry expert, Jordi Lanas, who is an immunologist and expert at all experiments pertaining to mice and acquisition of tissues and whatnot. And then we have Pragya and Hannah, who run our mouse facility, the mouse room itself, and do a lot of the bench work in the lab and help us out with a tremendous amount of work. And unfortunately, Pragya is leaving us tomorrow, so we are going to miss her dearly. Okay, so what do I mean by HIS mice? I will use the word HIS or humanized. It's actually easier to say humanized, but I refer to them as human immune system mice to differentiate them from other types of humanized chimeric mice, which may have a human protein or human liver bits or whatever. So these are human immune system mice. And basically, a humanized mouse is the injection of human immune cells into an immunodeficient mouse strain. And the mouse has to be extremely immunodeficient to be able to accept this human immune system. And there are lots of models out there, and I'm just going to provide an overview of the basics. And the easiest way to make a humanized mouse is to inject human PBMCs, blood cells, right into the immunodeficient mice. You can get the human cells will take, and you have a human immune system. We will talk a little bit later about the differences in the characteristics of these, but when I think of humanized mice, I think of the ones that are made from a human hematopoietic stem cell, a CD34 positive cell, which can come from several sources. We use cord blood from the Clinimmune Bank, but others use fetal liver as a source. Unfortunately, if you take urinized blood, and we can isolate our CD34 cells, there's plenty of them, they do not engraft and make a human hematopoietic cells. They aren't good stem cells. So you have to have a source of a very young human or from the bone marrow of an adult, which you can also get from the mobilized blood, from GM-CSF. However, that's harder, obviously, for us to obtain than a cord blood unit. And even cord bloods are easier than even getting a fetal liver, which has one source. So the other requirement, you have the stem cell source, you have to have the immunodeficient mouse strain. So basically, they started developing this model to study human HIV. Since HIV was a human-specific virus, they've been trying since the 1980s to get good engraftment of human immune cells into the mice and then be able to infect those human immune cells with HIV. So we knew all along you couldn't have the lymphocytes. The T and B cells had to be gone. And that is accomplished in various strains through different mechanisms. One of them is the SCID mutation, which is a DNA damaging mutation, which kills the lymphocytes specifically. And this is what we think of as the NSG strain, so the NOD. So when you have the SCID mutation, since it's DNA damaging, there's also radiation sensitivity issues that you need to be aware of. The other way to get rid of the T and B cells pretty easily is through the genetic mutation of the RAG1 or RAG2, which are absolutely required for VDJ recombination of the human T cells or the human B cells. And when you have a deletion or a mutation in either of those genes, you get a complete T and B null mouse. So then the big advent that came around in the early 2000s was the addition of the gamma gamma chain IL-2 receptor knockout, which allowed for NK deficiency. And all of a sudden we went from having humanized mice, chimeras with 1 to 5% engraftment to 60% engraftment. So it was a huge step forward in this mouse model. And through the years, we have discovered that there's one more tolerance we need, and that is macrophage tolerance that really enhances human engraftment. And this is controlled by the SERP alpha gene, which binds CD47. So the macrophages express a SERP alpha, and your other cells, including your red blood cells, express CD47. And when those are autologous, they bind each other, and that gives the message to the macrophage to not eat me, don't eat me. And it just so happens that the NOD mouse, the NSG strain, their SERP alpha binds human CD47. So that strain engrafted human cells quite well. Whereas other strains, including like C57, the strain we would love to have humanized mice on, does not bind human CD47 at all. And so you get terrible engraftment. The model we've been working with is the BALB-C, which was made early on in these whole development of the IL-2 common gamma chain receptor knockouts, and it binds human CD47 not as well as the NOD model, but it was working for humanized mice, and we used that for several years. And then once this SERP alpha contribution was known, several groups then tried to solve this on the BALB background by back crossing to the NOD. And so the mice we use have the NOD SERP alpha, but the rest of it is the BALB with the RAG2 mutation. And you could also do this, but other groups have solved this by transgenesis of the human SERP alpha. So that's basically the requirements for humanized mice. You have to have a source of human hematopoietic stem cells, and you have to have an immunodeficient host. And the way we make these is we, like all the groups, positively select the CD34 hematopoietic stem cells through magnetic beads from cord blood. And then we, our group expands them in culture typically for about 5 days. And then we're able to freeze these cells, and they actually work well. So we inject neonatal mice, so at day 1 or 2 after birth, just so that we can have them humanized for longer. And neonates are notorious for being more tolerant of a more tolerant immune system. So when pups are born, the next day or 2, we give them light irradiation and inject into their facial vein or into their liver. And then you wait, because as any bone marrow transplantation or cord blood transplantation, it takes weeks to months to develop this multi-lineage human immune system. And so theoretically, we get a multi-lineage human immune system, and we can generate from one cord blood, on average in our group, we generate 40. Other groups tend to generate around 15 mice, and that's just the source of your cord bloods. But we have generated up to 90 mice from one cord blood. So then you have a lot of mice you can do an experiment with. And to show you that not only the multi-lineage and multi-organ engraftment, we get engraftment, as you can see, in the lymph organs, lymph nodes, spleen, bone marrow, we get thymi. But we also see the human cells, and we detect these human cells by this hematopoietic marker that's on all immune cells, CD45. So those are the human, and we also always stain with the mouse. So we look in the blood, but then we look in the organs at the end when we harvest. And the one tissue that gets very low engraftment is the gut. And so we are working on trying to improve that. But we also can then look at the types of human immune cells in them. And if you wait, you have to wait several months to get robust T cell engraftment. But we do find by the CD3 marker, good T cells in the various organs and B cells. And you can tell that our model of humanized mice, like many other models, is largely lymphocytes. So when we say multi-lineage, it's skewed lymphocytes. So if you're studying lymphocytes, it's a good model. But we do get human myeloid cells. So if you look at those ones that aren't TRB cells, and you look by a variety of myeloid markers, we always include this positive control in our stainings, which is a human blood sample that's mixed with mouse spleens to represent the mouse plus human mix. But you can see we get nice populations in the various organs of the myeloid cells. It's just not as high a frequency as it is in you or I. And importantly, at our institute, we have shown time and time again that we do not see a sex disparity between how chimeric the mice are. So the donor is either female or male, right? But the recipients, we use both females and males. We inject them as a 1-day-old. We don't know what they are, but they're equally chimeric. And so what can you do? What kind of studies can be done with the humanized mice? And I'm going to give just a few examples of the types of studies we have run and talk theoretically about the types of work you can do. First of all, you can study human immune system development, which Robert and I spent a lot of time doing that. You can study responses to human viruses that are human-specific viruses, which is why this model was developed in the first place, and such as HIV, EBV. It has to be a virus that infects the human hematopoietic system, or you need to manipulate your recipient so that you get infections, such as they have made models to study SARS-CoV-2. Huge efforts now in the last 5 years have been brought forward to study human immunotherapies to human-specific tumors, including checkpoint blockade, CAR T cells, BiTEs, all the realm of immunotherapies that you see in clinical trials. Others have used the model to study autoimmunity in humans. And you can also, and I'll talk a bit about looking at the function of human hematopoietic genes. You can essentially make knockouts of these genes in this system to study them. So back to these different models. So why not just always just throw the PBMCs into the mouse? That seems so easy. You get engraftment within 2 weeks. But the problem is the blood cells from you or I are a mature human immune system that was developed in me. So I'm tolerant to me, but I'm not immune tolerant to this mouse host. So GVHD ensues in these models. There have been manipulations to extend this window, but typically you need to do the experiment within 4 weeks. And really, it's a model of very activated T cells, because the T cells are very busy reacting against the mouse. So they grow out, proliferate, expand, and you can get good chimerism, but you have very weak B cell, NK, or myeloid engraftment. But for certain systems, they're useful, and I'll show you how it's used for the cytokine release syndrome studies. Whereas if you make a mouse from the hematopoietic stem cell, the T cells are being selected in the mouse thymus itself, and so it's tolerant to the mouse and to that human donor as well. You don't get GVHD by and large, and you can get a more multi-lineage human immune system. And so your studies can be a much longer duration, which can be good and bad, but it takes months to do these studies. So just a few examples. So with the HuPBL, the Jackson Labs showed in 2020 how you can use this system to do very short-term, easy experiment assays to look for cytokine release syndrome, which is a toxicity of many of the immunotherapies, not the checkpoint blockade. That's a longer-term autoimmunity. But for like in putting a CAR T cell or a BiTE where you're engaging the T cells and they go through a cytokine storm, you can use this model to study your reagent, and you basically just wait a few days after engraftment. You give the drugs, plus or minus drugs. You can measure the body temperature, and literally just hours later, sacrifice the mouse, collect the sera, and with various assays, look for the cytokines, and you can find huge inflammatory signatures in these mice. So we have run one of these kind of studies with the group. You can also use this model, and we and others have done this, to look for this immune-related adverse events, or IRAEs, associated with checkpoint blockade inhibitors or maybe other immunotherapies. And so you can put tumors in or not, you give the drugs, and you can look with the model at lots of different things, including immune infiltration into various tissues, activation of the immune system, and autoantibodies. And we have a collaboration ongoing with Dr. Joy in pharmacology. And she sent a bunch of our samples that had been treated or not with a PD-1 or CTLA-4 inhibitors that are used frequently in the clinic now. And they looked at the kidneys in these mice and found that there was an increase in kidney pathologies by several markers, measurements, and this is one. that showed that with the treatment, in the treatment groups in the mice that had the human immune system, the controls were actually the same strain that were treated similarly, but did not have the human immune system. And so you could use this model to look at toxicities associated in this case in the kidney, but also in other organs or whatever you want to look at. As I said, we have, Roberta and I have been using this model to look at the development and selection of autoreactive B cells. And we have shown that actually we were surprised to find that the human T cells were very instrumental in the maturation of human B cells. And also by making a novel recipient, it's still immunodeficient, but it also harbors a transgene that is autoreactive to a subset of the B cells, human B cells, so that you can trace their fate in vivo, and found that although we get similar mechanisms of central tolerance, so the B cell doesn't develop well in the mouse if it's autoreactive, we found that there's a lot more variability based on donors than what you would see in a syngeneic mouse model, right? And we have done studies with human-specific viruses, along with Rosemary Rocheford's group, where she has been studying this other strain of EBV that is endemic in Africa and has been associated with T-cell lymphomas in children there. And they hypothesized that this strain was actually infecting the T cells. The kind that we get, EBV1, infects B cells and not T cells. And using this humanized mouse model, we are able to show that actually the EBV2 strain infected both the T cells and the B cells, whereas the EBV1 strain infected only the B cells. And I just want to bring up this concept because I think it's really a cool application of the model, that you can genetically modify the hematopoietic stem cell. So what you start with is the stem cell. You can do CRISPR or lentiviral transduction for modifications of a gene of interest that is in an immune cell, change it in the stem cell, and then you have the ability to develop a human immune system in mice with or without that genetic modification. And we have done just limited amounts of this, but we are working with Dr. Fujita on optimizing doing CRISPR modifications of these stem cells, but it's a kind of cool way to just study a human gene of interest in a in vivo model. And I will then be talking the rest of the time basically about the use of these mice for immunotherapy studies in vivo using a humanized mouse model. We have published 4 papers with different collaborators and our own, showing how we can look at the human immune response and how it correlates to tumor regression or a response. And we have the 5th paper in press right now. So as I said, this is a high use of these mice currently. And why would we want to use the humanized mouse as opposed to just doing a clinical trial? Well, you have, it's cheaper for one thing. You can run studies with and without treatments and get a lot of data more quickly. And why not, instead of a syngeneic mouse model, I always think it's in addition to the syngeneic mouse model, because you now have the variability of human tumors that you can study. You can put PDXs in patient-derived xenografts into these mice and grow them. And so they can have the huge variability that human tumors really do. And you can study combination therapies. Oh, the immune system is human as opposed to mouse, which there are differences, which we have found in some clinical trials when we've gone to clinic and found that the reagent was actually toxic to humans, and whereas it wasn't in the mouse. And importantly, you can get a lot of tissue, so you can do a lot of studies. And I'm just going to give a few examples of the type of experimental possibilities you can do. Like you can look at the kinetics. You can look at whether you give the chemo before or after the immunotherapy. You can look at different dosing. You can do a lot of flow cytometry, even mass cytometry, because you get a lot of tissue, a lot of tumor tissue and lymph nodes and spleen to represent the systemic immune system. You can prepare some of these for downstream IHC or histology. You can prepare some of the tissue for RNA. Importantly, you can do mechanistic studies in vivo. You can add antibodies where you can block or not certain populations that you think are important in a response. As I talked about, you can genetically modify the stem cell. Also, you can do genetic modifications of cancer cell lines to test whether that is important in the tumor microenvironment. And I've talked about these other immunotoxicity studies. So what do we offer? I'll go through what we do, and I'll finish quickly with examples of how we have done these immunotherapy studies. So importantly, you can buy humanized mice from other groups. We offer them cheaper than other groups, significantly cheaper. But we are also immunologists. And so in almost all studies, we also run the immune analysis. We harvest the tissues, we do the flow cytometry, we do the analysis, we present a report. So cost comparison of these humanized mouse models. So you can get these commercially from several institutes, and I'm giving a couple examples, and they run between $900 to $1,200 typically for a mouse that's been tested once for its human chimerism with a minimum 25%. We offer them for around $370 and $1,400 a mouse for the full flow analysis. And I will talk in a little bit about what that includes, and all of that can be optimized, adjusted, and the price would be adjusted, of course. I've also put in a couple of these other strains to represent that there are a lot of other strains always being developed that are improving on NK function or myeloid function. And so those can be purchased. And if we're running them in-house, we can make the models for you, make the humanized mice and run the models. So we also know how to do the tumor injections, tumor measurements, dosing, and all that. So the workflow, just to get this out of the way, the easiest way to get involved is to contact me or Roberta, and here are our emails. And we will then discuss, I will then discuss with you the feasibility of the project, the timeline. We are always making humanized mice, and so we have a wait list always, and I can tell you how long it takes. Remember, it takes 4 months to even get the T cells. So it's, you know, at least a 6-month out projection. We provide the overview. The investigators need to add the mice to their protocol. And what we do is we bleed the mice twice and make sure that they are chimeric. And that is important so that you reduce the variability in your investigation, in your experiments. Our mouse are housed in the barrier, and so it's a restricted access. So if you want to perform your own experiments, we can just export our mice to your rooms, and it doesn't require any quarantine because we're in the barrier. Or you can have us run the studies in our room, and we would do the work because we're the ones with the access. So we're flexible. We work with you on different things. If you want us to do the harvest at the end, which we often do, if we want to do it with you, if you want the cells afterwards, we will work with you however you want to run that. But we often run the flow cytometry, we prep the tissues for histology and whatever. So in the last couple of minutes, I'm just going to talk about our protocol for testing immunotherapies. We've run over 40 experiments of these now. We often run combination immunotherapies. So they're testing drug plus a checkpoint blockade. Or you can test CAR T cells. You can test whatever immunotherapies you want, and we would adjust the protocol. But basically, you know, we make the humanized mouse. The little baby gets injected, it gets weaned from its mom, and when it's 10 weeks old, we bleed it and look in the blood for the human cells. And we establish the rate of chimerism in this mouse at both 10 and 15 weeks, because it can change over time. And at 15 weeks, it gives us a better idea of how many T cells are there. That can be the biggest variable. That is the biggest variable among different cord bloods and among different labs, is how many T cells you get. And if you're studying T cell immunotherapies, you want to be sure you have the T cells. So with that information, we advise your groups. So if you're doing group A, B, C, D, E, we try to make them as equivalent in human and T cell chimerism as we can to reduce that variability. We advise when to inject the tumor based on the tumor growth characteristics. And when the tumors are around 150 cubic millimeters, treatments are started. And at the end of the study, we are, in almost every case, we are involved in harvesting. We like to collect the lymph nodes, the spleen, and the tumors themselves. And then we prepare those into single cell digest for flow analysis. So importantly, any tumor, we have never found a tumor that grows in a nude that does not grow in our mice. And as a matter of fact, they usually grow better in our humanized mice. And that we can find evidence in some tumors respond to just PD-1 blockade. And so we can see that in the mice that harbor the human immune system, we can get a difference in the tumor growth based on the immunotherapy. But importantly, you don't see that difference in the control mice that do not have the human immune system, but are the same genetic background. So we always have these mice around to run as controls as well. And that this growth rate is very similar between the humanized and non-humanized. And also very importantly, is that the amount of immune cells, human immune cells, and even mouse immune cells that get into the tumor is tumor dependent. We and others have shown this, that the tumor microenvironment of that specific tumor is dictating how many cells go into the infiltration into the tumors themselves. And we see this by flow cytometry. So you can see some are hot and some are cold and some are different even among the same type of cells. So we propose that this maintains its basic tumor microenvironment that's dictating how infiltrated the tumor is. And that's very important, because if we just treated all these tumors, any old tumor, and got the same response, the model wouldn't be worthwhile. But we are finding a lot of differences. And we can see these differences. We inject in both the right and left flank. These can be done orthotopically. There are people that put brain tumors in them. But when we do the flank model, the amount of infiltration that goes into each mouse is very similar. Each dot, the colored dots are the left or right, and they can be variable. But that's independent of how big the tumor size is. The tumor sizes can be very different from the two different sizes, but how infiltrated it is very consistent. From this data, we look at tumor growth, which we've done a series of colorectal cancers, all of the same, they're all kind of the same type, but some of them responded quite well to the, and the measurement we use is so that we can look at the growth rate over time, because sometimes treatments are started on different days and we harvest on different days. So by this measurement, some tumors respond quite well and others do not. And we think that actually, we are trying to get more clinical data to say that represents this physically. Importantly, we do a lot of flow cytometry. We look at the human immune system, both systemically in the lymph nodes and spleen and in the tumor themselves. And we look how activated these T cells are, how many myeloid cells are there. We look at whether the T cells can produce cytotoxic molecules like granzyme B and interferon gamma, TNF alpha. This is an example to show how activated. You can look at effector memory T cells. You can look at inhibitory receptors. We have lots of Tregs in this model that we can quantify. And again, we can look at these cytotoxic molecules. And all of these assays can be customized. And more importantly, we can even look at the tumor itself by gating on the cells that are not the immune cells and having a tumor-specific marker when we can look at things like the expression of class 1, class 2, PD-L1, and see if these markers change with treatments. And we have found in some cases it is, and in some cases it doesn't. And we can correlate any of these measurements to that tumor growth. And here we show that in one model, the more T cells there's more T cells in the smaller tumors, which makes sense. And that more of these T cells are producing interferon gamma. So we can send them off to Vectra and look at the spatial organization of these immune cells. And this is honestly my last slide. So I'm just trying to show you that there's a lot that can be done with these models, that all of these, this can all be personalized. And there are ongoing worldwide efforts for improvement of the HISMOS models by others and ourselves always to try to make it more physiological.